So in Chapter 20, we're going to get your introduction to antibiotics, so antimicrobial drugs. Okay, so I don't even know if I should keep this slide anymore. So a chemotherapeutic agent, okay, aka drugs, is anything that you can take to treat something wrong with the human body. Okay, so drug is a very, very broad term. Okay, you can take drugs for all sorts of syndromes and disorders and genetic disorders or diseases. Okay, it's a very, very broad spectrum term. So in this chapter, we're focusing again on antimicrobial drugs. And we are going to really, really focus on antibacterial antibiotics. Okay? The bulk of the chapter is talking about how to kill bacteria alone, which will clear up, hopefully, a lot of misconceptions. Because at the very end of the PowerPoint, there's a few footnotes where we address antivirals, antifungals, and antihelminthics. Okay? They're drugs unto their own. Okay? Most antibiotics are just antibacterial. So, chemical substance that destroys disease-causing microorganisms with minimum damage to the host. That's another thing we're going to clear up. Okay? Antibiotic, anti-life drug. When you take antibiotics, while they do effectively kill bacteria in your body, they also do a certain amount of damage to yourself as well. People don't often realize that. All right, another misconception. Many folks think that antibiotics are fully synthetic, man-made. That's really not the case. Some are, okay, few and far between. Okay, the classic ones are sulfa drugs and trimethoprim. Maybe you've heard of Bactrim. Okay, those are synthetic. But most antibiotics are actually naturally derived. Okay, we find microorganisms that produce them naturally, and then we exploit those organisms. We say, hey, you're making this fabulous antibiotic. Teach me. And then we take it, we see how it works, and then these days we make them semi-synthetic. We say, hey, this new antibiotic, it's pretty effective. Well, let's see what happens when we chemically tweak it. And then it turns out it's even more effective. So we'll look at that later on as well. All right, so since it's spring and I get a whole extra day in the spring, I want to take the couple minutes here to talk about a little bit of historical microbiology. I love this stuff. And talk about the first two major antibiotics. Sulfonamide, or sulfanilamide, same thing, sulfa drugs, and then penicillin. Okay, so first scientist we're going to address here briefly is Paul Ehrlich. Okay, early 1900s, Paul Ehrlich was trying to find the proverbial magic bullet. He wanted to find some type of medicine that would be effective at curing all microbial diseases. And he was really focusing on syphilis. Okay, so syphilis these days is generally not a big deal. Okay? Easily treatable with antibiotics, but prior to antibiotics, syphilis was very problematic. If left untreated, you were potentially left with a new little friend inside of your body for the rest of your life. And syphilis can lead to a lot of severe lifelong disorders. Okay? You can die from neurological decay because of syphilis. You can die from heart failure because of syphilis. Your own skin can consume itself because of syphilis, which we'll see the very last day of the semester. I got beautiful pictures for that day. So he wanted to try to fix that. And the first drug he came up with was called Salvarsan. This is an arsenic-based powder. Okay? It's quite poisonous, so it would make you feel awfully sick, but it was pretty effective. And if you're not familiar, arsenic is an inhibitor of respiration, so it inhibits electron transport, which is why it worked pretty well. But it's also why it's poisonous. So that led to Gerard Damiac, who said, hey, Ehrlich's got this Salvarsan, it's kind of effective, let's see what I can make. So Gerard Damiac was trying to treat Streptococcus pyogenes, strep throat. And like syphilis, if strep throat is left untreated, it can lead to a lot of lifelong disorders. Rheumatic fever, glomerulonephritis. Okay, you can die from heart failure from untreated strep. So he came up with Prontosil. Okay, it was this red dye that was also potentially an effective drug. When I say potentially, there's more to it. So Prontosil started off as a failure. In vitro, okay, because you got test on test tubes and petri plates, right? Prontosil was not effective. It didn't do a thing to strep pyogenes cells. But he gave it to individuals anyways because, hey, no FDA. And when Prontosil was given in vivo, it was highly effective. What the hell happened, right? It was useless in a test tube, but when somebody took it, it was great. Turns out Prontosil was a precursor. When you take Prontosil, it's metabolized into sulfonamide. Okay. We've actually talked about sulfonamide in the past when we discussed competitive inhibition of enzymes. And we saw that one example with the sulfa drug that binds to the enzyme that makes folic acid. So, this was highly effective because the sulfa drug mimics PABA and prevents bacteria from making folic acid, killing them. Okay? Doesn't really harm us because we don't make folic acid, we eat it. So we don't have to worry about that enzyme. So the sulfa drug was the first successful and prescribed antibiotic. Alright, that brings us to Alexander Fleming. Most folks know this dude because it's the guy who discovered penicillin accidentally. So, story goes, he was researching staphylococcal bacteria, and one of his plates became contaminated. Became contaminated with a nice, fuzzy fungus colony. And what happened was that around that fungal colony, there was a clearing. Okay, and the rest of this, over here, this is staph. He thought that was kind of weird. You got all this staph growing, and then you got this no man's land, and then you have the fungus. What was preventing the staph from growing in this zone around the fungus? Well, he looked into it, and it turned out the fungus was producing a molecule that was killing staph, penicillin. So he's the guy who discovered the organism that produces penicillin. Okay, Aspergillus penicillium. The two dudes who did something amazing that often get left on the wayside are Chain and Flory. Okay, Fleming discovered penicillin, but more importantly, Chain and Flory figured out how to mass produce penicillin. Okay, this was early 1940s. Okay, the thick of World War II. This helped save a lot of Allied soldiers. Okay, so their efforts to make penicillin were so successful that penicillin was so widely available you could buy it over the counter. Okay, people took it every damn day if they so chose, but there was a problem. Resistance developed within 5 years of their figuring out how to mass produce it. And that was the beginning of a story that's becoming a major issue these days. Okay? Abuse of antibiotics is leading to a lot of widespread resistance by bacteria. All right. That's my little background there. So getting back to the good stuff. So like I mentioned, most antibiotics these days are naturally derived. So where do we go looking? Beneath your feet. Soil microbes have provided us some of our best antibiotics. It makes sense when you think about it. So in the soil, you have, pound for pound, more bacteria than any other place on the planet. Okay, and this is topsoil, mind you. So you have all these bacteria crammed together. That's going to lead to competition, right? Nutrients, resources, real estate. And how do they compete with each other? they will produce antibiotics. So, we look to bacterial species like Streptomyces. Streptomyces have given us some of our best antibiotics. And Bacillus species, like old friends like Bacillus cereus or Bacillus subtilis. These bacteria naturally produce a wide variety of antibiotics. And then a number of fungal species do as well. So, those in the species Penicillium or Cephalosporium have been particularly handy. Now, there's a bit of a problem. The number of new antibiotics we're discovering has dwindled massively over the last 40 years. Okay, so as you can see in the bar graph, as the bar graph goes down, that represents the number of antimicrobial approvals. So, to make this even more sobering, next time we're going to talk about the 5 classes of antibiotics. Each one is defined by what aspect of growth it inhibits. There has not been a new class of antibiotic in 40 years. Okay, so we're running out of ideas as bacteria learn how to be more and more resistant. I'll skip this. This is just showing you folks where a lot of popular antibiotics are derived from. Alright, so I want to do first 2 slides here and then I'll shut up. Okay, so we're going to start talking about the features and characteristics of antibiotics. Okay, so the first characteristic, selective toxicity. Okay, so antibiotics are selectively toxic, meaning that ideally, when you take them, they will do more damage to the pathogen than to you. Okay, so they're all toxic. Okay, they're all going to do a certain amount of damage to you, but it would make more sense if you have an antibiotic that does more damage to the pathogen. Now, why they're selectively toxic will make more sense next time, but antibiotics target growth characteristics common to bacteria and different than in humans. Okay, so common to bacteria, but not to humans. So, there's a lot of antibiotics out there. And they do vary in their selective toxicity. Okay? Some are a bit more harmful to you than others. So, how do you measure that? Well, that's therapeutic index. Okay, so forgive me, I always have to read this one. So to calculate that, you need to know the lowest dose toxic to the patient. So how much can you give somebody before it hurts them? Divided by the dose typically used for treatment. Okay, so big number divided by little number. So you end up with an inverse scale. So a high therapeutic index is ideal. So the closer you get to 10, the gentler the drug is on you. The closer you get to 1, though, so on the low side, those are going to be more damaging to you. Okay, so I'll reference this as we go on. So high is good, low is bad. Alright, so next characteristic. Are the antibiotics bacteriostatic or bactericidal? So many antibiotics are merely inhibitory. Okay? They don't outright kill the cells. Instead, all they do is slow them down. That's bacteriostatic. So antibiotics are the proverbial wrench in the gears. Okay? They'll stop some of the gears, but not all of them. So, these are ideal because they tend to have a higher therapeutic index, a little gentler, and they're good when you know what you're dealing with, so the immune system can catch up. But say you have a pathogen that you're unfamiliar with, particularly aggressive, then you may have to rely on bactericidal drugs. These don't just slow growth, they stop it. These guys outright kill the cells. So they stop all the gears. Okay? Many of these can be a bit on the low end of the therapeutic index, a little bit more damaging. We'll pick up here next time. Alright, so we'll finish this up next time without issue. May start a little chapter 16. We'll see. But otherwise, folks, I'll get the quiz posted for 20 here shortly. Remember the one for chapter 13? That quiz is already posted and due Sunday. But otherwise, I'll see you in lab for day 3 of the unknowns. Spectrum of activity. Okay. Antibiotics target different aspects of microbial growth, which means not all antibiotics are going to work against all bacteria. Okay, so you have to consider which organisms will actually be affected by said antibiotic. So many antibiotics are narrow spectrum. Okay, they only work against specific groups of bacteria. So traditionally, you'll see them effective against either gram-positives or gram-negatives. Okay, not both. So penicillin is highly effective against gram-positives, doesn't do anything against gram-negatives. Now on the flip side, you've got polymyxin B, used in a lot of topical antibiotic ointments. Okay, polymyxin B is great against gram-negatives, but it doesn't do a thing against gram-positives. So these are a little bit more useful when you know exactly what pathogen you're dealing with. Okay, and ideally these are a little bit gentler on the human body. Okay, these tend to have a higher therapeutic index, but not always. All right, now the scary ones are the broad spectrum drugs. Okay, these are becoming your drugs of last resort because they're effective against a great variety of bacterial pathogens. Okay, these tend to be a bit more on the toxic side. Okay, so they're useful though when you don't know what kind of pathogen you're dealing with, or a pathogen is not responding to the typical antibiotic treatment. Now, these do have one major drawback, besides being slightly more toxic. You have to consider what happens to your, say, gut bacteria when you take antibiotics. Okay, you're loaded with bacteria. Even if you take narrow spectrum drugs, do you think that those antibiotics aren't going to do anything to your gut bacteria? Or is it probably going to bring the numbers down? It's going to bring it down, folks, right? They're antibiotics, right? They work against bacteria. The problem with broad spectrum drugs is they're a bit more hard on those gut bacteria. So it is not uncommon to have a doctor say, all right, I'm prescribing you this antibiotic, go buy a probiotic to go with it. Okay, that's becoming very commonplace. Okay, so like I said, antibiotics vary greatly in their spectrum of activity, which you try to illustrate here. So 5 groupings of bacteria. You know, we know gram-negative, we know gram-positive. We've seen mycobacteria, tuberculosis, for instance. The chlamydias and the rickettsias, they reproduce in slightly unusual ways. I don't get into it in this level of microbiology, so they get lumped into their own categories. So you can see some are very broad spectrum, like tetracycline. It's great against just about everything. Penicillins, these days are kind of in between. They're what we call extended spectrum. Great against gram-positives, but they can actually affect some gram-negatives these days. And then, if you go over to the Mycobacterium, the red column, at the bottom you've got isoniazid. The only time you'd ever take isoniazid is if you had tuberculosis. Okay, this is a very, very narrow spectrum drug that is only effective against those waxy mycobacterial cells. Okay, wouldn't do a thing against anything else. You have to take into consideration what happens when you take combinations of antibiotics. Okay, so this is commonplace in hospitals where somebody may have an aggressive infection. They're not just going to give you one intravenous antibiotic, they may give you several. So ideally, when you use antibiotics together, they are synergistic. They boost the effectiveness of each other. Okay, I'll have a great example later on with sulfa drugs and trimethoprim. Now, apparently, and I've yet to find a really good example, they can be antagonistic, meaning the antibiotics would hinder each other, but again, I have yet to find a decent example to give you. So apparently it can happen, but I don't know really of a good applicable example. And then in some cases they're just additive, where you take this combination of antibiotics and they're just kind of there. They're not hurting each other, they're not helping each other, they're just kind of doing their own thing. But you do have to take care in considering how will these antibiotics interact with each other? Next feature to consider, okay, tissue distribution, metabolism, and excretion of the drug. Okay, so when an antibiotic gets into your body, for one, will it get to the site of infection? Okay, so some antibiotics you can just be taken orally, they dissolve in the bloodstream, they can spread everywhere. Okay, and that's for more superficial infections. But what about, for instance, meningitis, an infection of the central nervous system? Here you've got a big problem, the blood-brain barrier. Okay, triple membrane protection. Most antibiotics cannot get through the blood-brain barrier. So how do you get at an infection there? Well, you have to use something like rifamycin. Rifamycin is one of the few antibiotics that are non-polar enough that they can pass through the blood-brain barrier. Okay, so you have to consider if I give them this antibiotic, will it get to, say, the central nervous system? And then some are just pH unstable. They cannot be taken orally, so they have to be administered intravenously. Rate of elimination. Okay, anything you put in your body is on the metabolic clock. Okay, when antibiotics get into your body, eventually they get scrubbed out by your kidneys and urinated. So antibiotics vary wildly here. And we measure this as half-life. Okay, and like the half-life of anything, which is the time it takes for the half of something to disappear, this is the time it takes for the half of the dose to be excreted. So this is actually a very important calculation because this is used to determine the size of a dosage and how frequently you take that dosage, right? It's good to know how long it's going to be in your body when you're administering an antibiotic. So like I mentioned, they vary wildly. One of the original antibiotics, penicillin V. Okay, penicillin V could be taken orally, but had to be taken every 6 hours. Okay, that's 4 times a day. Fairly heavy doses. Now, on the other hand, we get azithromycin. That's the Z-Pack. Right? Most of us are familiar with the Z-Pack. And what? You take 2 the first day, and then what? 1 each day after that? Okay. Frequency of dosage is 24 hours. That's a big difference. 6 hours versus 24. A few years back, I had one poor student. She had this terrible ulcer in her eye. And the infection was so aggressive that she had to carry this ice chest with her of several different antibiotics, and she had to leave the room every 20 minutes to apply eye drops. So that is the most severe dosage I've ever seen, but just to show you, they vary wildly. Incidentally, her eye recovered with just very minimal scarring. I was amazed. All right. So next thing to consider, adverse effects. Right. So antibiotic, anti-life drugs. Okay. Not just to the bacteria, but also to you to a lesser extent. So toxic effects. This is side effects. Right. You watch a pharmaceutical commercial of any sort, and kind of thrown in the middle, they list all these horrific things that could potentially happen to you. Antibiotics are no different. Okay, so what sort of rare side effects may you encounter? So looking at some drugs of last resort. Streptomycin. Streptomycin can cause permanent kidney damage. It can be ototoxic, where it can damage your inner ear, leading to impaired balance and deafness. I actually have a story to go with that in a second. Chloramphenicol is like the drug of last resort these days. In very rare cases, this is a scary one, it can cause aplastic anemia. Your body loses the ability to make blood cells. Okay, that's pretty severe. I looked into it. Apparently it is reversible if it's caught early and survivable. Others that I don't have space for, I gotta look up the name of it again. About a year and a half ago, 2 years ago, my wife has a penicillin allergy, so they had to give her something else, and I had never heard of it. And I looked it up, and one of the rare side effects was hepatic liver failure. Oh my God. So we were like, hey, can you give her anything else? We're like, okay, well, there's this newer beta-lactam out there that is actually safe to take for people with penicillin allergies. So I'll mention that again in a second. And then my fun story, so just over a year ago, so was it last February, I was prescribed the fluoroquinolone. I had an infection in my leg. And within 48 hours, I was lecturing, and my ear started screaming. Like it was ringing like I'd never felt before. I was getting dizzy, and the other ear started ringing. And I'm like, okay, what the hell's going on? I'm like, okay, stop taking it. Okay, it's ototoxic to you. I'm like, what does that mean? I'm like, well, it's damaging your inner ear. Let's see. Right now, I can still hear a little bit of ringing. I get permanent tinnitus because I took an antibiotic for 48 hours that didn't agree with me. That spring semester, I sat my butt on that table the whole semester because I was dizzy for the better part of 6 months. It was fabulous. But eventually got over it. I still get occasional dizzy spells when the weather gets really wonky, so that's fun. And then allergic reactions. Okay, your immune system can choose to not like anything it wants. Right? That's an allergy. You become hypersensitive to some organic molecule. So allergies to antibiotics are like allergies to anything else. Okay, some people have fairly mild reactions. My wife, when she takes penicillin, or most beta-lactams, she breaks out in hives. Okay, but like I mentioned, they actually have a newer class of beta-lactam that people with allergies can safely take, which was amazing. She took it without any reaction. And then other folks can have severe allergies, anaphylactic shock. All depends on the individual. And then the last adverse side effect is actually something that we're researching heavily right now. What happens to those gut bacteria when you take antibiotics? Okay, we want to know how strongly are they impacted. Because as we prescribe so many antibiotics, we're running into problems like antibiotic-associated colitis. Someone took antibiotics for so long, or at high dosages for long enough, that they killed off their colon bacteria to the point where something like Clostridium difficile can move in. So I know several of you folks are probably familiar with C. diff. Okay, every one of us has C. diff in our colon. It's kept in very low numbers. You have actually a lot of Clostridia in your colon. It's just kept in check. The problem is with this form of colitis, the antibiotics kill off the good bacteria to the point where C. diff says, hey, I can move in. Okay? C. diff is multi-drug resistant. It's very difficult to treat with antibiotics. And when it takes over in the colon, your immune system isn't happy about that. So you get chronic inflammation, the organism causes some tissue destruction. In the past, the only good way to treat it was segmentation. Okay? Remove the portion of the colon afflicted with C. diff. These days, though, and I'll talk about this more in the last week of class, there's newer treatments like fecal transplantation, which sounds horrific, but has been wildly successful. Where they can take feces from a related family member, transplant it into your colon, and that has actually helped the microbiome fully recover. Okay, your poop is effectively time capsules of your microbiome. So, as gross as it sounds, it is a wildly successful therapy. Okay, so any questions about any of these features before we move on to the good stuff? All right, so this is my favorite part of this chapter because this clears up a lot of misconceptions. We're going to talk about how antibiotics actually work, right? What do they actually do to a bacterial cell? So here's the thing. A couple things to keep in mind. There are 5 classes of antibiotics. And each class is defined by what feature of growth is inhibited. So with that in mind, I want you to think as we go through this, is there any part of growth in a bacterial cell we have not touched? Okay, so just think about that as we go. All right, so the first and most popular class of antibiotics, those that inhibit cell wall synthesis. Put it another way, antibiotics that inhibit peptidoglycan synthesis. Now, this is a great group of antibiotics because they have generally a high therapeutic index, very selectively toxic. Like, we're targeting peptidoglycan here. Okay, is there peptidoglycan in human cells? No. So this is something that is very distinct to bacteria that we're trying to target here. Now, if you ever take a pharmaceuticals class, they're going to go through like tons of examples, and you have to compare and contrast. I'm only going to hit on a few examples with each. Okay, so for here, I want to talk about penicillin, vancomycin, and bacitracin. Okay, so let's talk about how penicillin works. Okay, and really, we're going to talk about how what's called a beta-lactam antibiotic works. Okay, so beta-lactams, penicillins, they all do the same thing. So way back when, we drew peptidoglycan like this. Right, we have all these glycan chain layers, those NAG and NAM layers, if you remember that. And then between the layers, you have that glue, those tetrapeptide bridges, right? Those amino acid bridges. So this is what penicillin does. It targets the enzyme that makes these bridges. So, the glycan chains are still made, but you don't get any bridges in between. So, that's probably going to weaken the cell wall pretty severely, I'd imagine. Take a look at the pictures. So, on the left, we've got bacilli, which are perfectly fine. But to the right of that, you got this mangled-looking bacilli. That one's been treated with penicillin. The cell wall has been weakened to the point where it burst. It lysed. All right, let's look at vanco. Okay, vanco's actually a little lower in the therapeutic index. That one's a little rough on the human body. But vanco is very straightforward. It just prevents glycan chain formation. So no NAG and NAM bonds, no cell wall. Now, bacitracin is kind of a gray area. Right? This is one you don't ingest. It's a topical cream. It's because it actually binds to the membranes of the bacteria. And what it's doing by binding to the membranes is it's preventing those NAG and NAM subunits from getting out of the cell. So if they can't get at the building blocks for peptidoglycan, they can't make a cell wall. So these are all in the same class, right? They all inhibit peptidoglycan, but they do so in very different ways. Right? Penicillin prevents the formation of the tetrapeptide bridges. Vanco breaks up NAG and NAM bonds. Bacitracin prevents NAG and NAM from getting out of the cell to make the cell wall. So, that's better. Why is my clicker not working? So you can go into great detail of all these different antibiotics, right? You can tell stories about all of them. I only want to spend some time on one group out of everything. Beta-lactams. Okay? The penicillins are actually a really interesting story. So to understand beta-lactams, first let's draw a general structure of a beta-lactam antibiotic. We're going to keep it ultra simple. So we start with a four-sided chemical ring. Okay, so in chemistry, rings can be triangles, squares, octagons, whatever. Attached to that, you've got a five-sided ring. Apparently, I can't draw. Okay, so a little house with a garage, and then you get a side chain. So beta-lactam antibiotics are somewhat similar to this, but what they all have in common is this square ring. That's your beta-lactam ring. Okay, so as we go through this story, remember this ring. And all of the beta-lactams do the same thing. They block the formation of those tetrapeptide bridges. So the first naturally occurring antibiotic we discovered was penicillin. Okay, penicillin is a beta-lactam. So here we've got penicillin G and penicillin V. Okay, first two discovered being produced by the fungus Aspergillus penicillium. So take a look at the structures here. So this is what I was roughly drawing on the board, that common nucleus. There's that beta-lactam ring, and then here's the side chain. Penicillin G had one big drawback. It had to be administered via injection. Now, penicillin V, on the other hand, could be taken orally. So that's our first oral version of penicillin. So what's the difference, right? You look at the structure, it's almost identical, except that one little oxygen sitting there. That one little oxygen atom makes a difference between whether or not penicillin can survive the stomach. So organic chemists figured this out. They're like, hey, what if we take these antibiotics and we chemically modify them further? Right? Organic synthesis, everybody loves that. So that's when we get into some of the first semi-synthetic penicillins. So the common nucleus is still intact with these, right? This is unchanged. We've got oxacillin and ampicillin. So the side chain of oxacillin is very much different. And by modifying it in this way, we made it resistant to penicillinase. Okay? This is an enzyme we're going to bring up later on. But this one could be administered to bacteria that are resistant to penicillin. And then ampicillin is still pretty common these days in lab settings. We're going to use ampicillin today in lab. Ampicillin, again, semi-synthetic, is our first extended spectrum beta-lactam. It was the first one that worked not only against gram-positives well, but against some gram-negatives. Okay? Most gram-negatives are very resistant to beta-lactams because of their outer membrane. And then the story goes on. Okay? Up until a few years ago, we went through 4 generations of beta-lactam antibiotics. And you can just see, as we go through these generations, the chemical modifications get more and more severe. Right? The side chain continues to be modified, but then so does the nucleus. The only thing that remained untouched was that beta-lactam ring. Right? That's the only part of the whole structure we never modified. So like I said, up until a few years ago, cefepime was the end-all, be-all beta-lactam. But then we ran into a problem. MRSA. MRSA, if you don't already know, stands for methicillin-resistant Staph aureus. Normally, Staph aureus dies if you say the word penicillin to it. Okay? MRSA, it's not fair to call it just methicillin-resistant. A much better description is beta-lactam resistant. Okay, so take your pick. Out of 4 generations, not a one of these works against MRSA. So MRSA continued to spread. We were relying on more toxic drugs to deal with it. So the FDA fast-tracked some new 5th generation beta-lactams. Okay? I actually had to change this diagram about a year ago because the one I had failed the FDA clinical trials. So this is the only one that succeeded in the U.S. to pass FDA trials. Okay? There's actually 2 others out in Europe. But ceftaroline, I mean, look at this monster. It looks nothing like penicillin originally did. This is the only beta-lactam in the U.S. that works against MRSA. Everything else is totally ineffective. Why that's the case, we're going to bring up in a little while. All right, so we spend more time on class 1 than any of the others, so any questions? Okay. All right, so the next class, class 2, are antibiotics that inhibit translation. Protein synthesis. So to put it another way, antibiotics that interfere with ribosomes. So that's obviously a really good target, right? Because if you remember, bacteria have 70S ribosomes. Humans have 80S. They are different enough in structure that the antibiotics, for the most part, prefer the 70S ribosomes. Now, there are a great deal of examples in this class. I only have a few, though. I just kind of want to show you how they work. Okay, so we'll start by breaking them into 2 overall groups. 30S inhibitors versus 50S. Okay, so these tend to target one of the 2 subunits of the ribosome.
All right, so let's see. Tetracyclines. Okay, so tetracycline here binds to the 30S portion and prevents the tRNAs from binding to the codon. Okay, so they prevent anti-codon-codon binding. So, if there's no tRNAs binding, there's no protein to be made. Streptomycin is in a group called the aminoglycosides. Streptomycin and its brethren bind to the 30S portion, and this is kind of clever. They prevent the ribosome from finding the start codon. It effectively blinds it. So if it can't find the start codon, it won't know where to start translation. Chloramphenicol, which is, as I mentioned before, a very potent antibiotic. It's in the family called the amphenicols. Chloramphenicol will bind to the A site and prevent peptide bond formation. So the ribosome cannot perform its enzymatic function. So, no peptide bonds being made, no protein being made. Now the last one, and you're like, hey, there's nothing there for azithromycin. I throw in azithromycin because, one, it's very widely prescribed. Most of you have probably taken the Z-Pak at some point. But I check every year, and we have still yet to figure out the exact mechanism of action. We know it screws with translation. We know it binds to the ribosome, but we still don't know exactly what it does to the ribosome. So that just makes me laugh because of how widely prescribed it is. Oh, and I throw the structures here just to show you, they look very different than beta-lactams. Okay? They're very distinct from each other. All right, the next group, class 3, antibiotics that inhibit nucleic acid synthesis. So that means we're targeting two different things. One, DNA replication. Right? We want to prevent the chromosome from being copied. And two, inhibiting transcription. We want to prevent the cell from making mRNA. All right, so fluoroquinolones. That's the one that I don't like. These are antibiotics that inhibit DNA gyrase. So here's the deal. Right? I inundated you with more than enough enzymes back in chapter 8, right? Primase, DNA polymerase 3 and 1, there's a whole list. I left out gyrase. So gyrase is an enzyme that relaxes DNA. Right? In all cells, DNA has to be tightly wadded up, supercoiled. Gyrase removes the supercoils so chromosomes can be copied. I don't know if your Gen Bio teachers talked about this, but the human equivalent is called topoisomerase. It does the same thing to our chromosomes. It relaxes them. So these enzymes, like ciprofloxacin and moxifloxacin, they bind to DNA gyrase, preventing the DNA from being relaxed. If it can't be relaxed, it cannot be copied. Okay, so it prevents replication of the chromosome within bacteria. The next one is rifamycins. And if you're taking rifamycin, it means you either have tuberculosis or meningitis. Rifamycin binds to RNA polymerase, and it blinds it. Okay, RNA polymerase has to find promoters on its own. Rifamycin prevents RNA polymerase from finding promoters. So if it can't find a promoter, it cannot transcribe genes. So they inhibit transcription. It is a unique antibiotic because it is incredibly nonpolar. It's one of the very few that can be used to treat central nervous system infections. It can cross the blood-brain barrier, as we mentioned earlier. It does have a common and unfortunate side effect called red man syndrome, meaning everything about your body becomes red or orange. So your skin becomes completely flush, your saliva, tears, and urine become bright orange, and you just generally feel worse than you already do. Okay, so that's inhibition of nucleic acid synthesis. So the next class seems like it should be the most obvious one. Antibiotics that inhibit metabolism. right? That just seems like the way to go, right? Inhibit glycolysis, inhibit Krebs, inhibit respiration, something like that. That'll kill a cell. Here's the problem with metabolism. It's that chapter that keeps showing up in like every biology class. That's because glycolysis is glycolysis is glycolysis. Krebs is Krebs is Krebs. These are highly conserved pathways amongst all organisms. So if you screw with glycolysis in bacteria, it's going to hurt you just as bad. So this presents a unique challenge. There are a few examples here, because you need to find some metabolic pathway that is essential for growth, but not found in humans. So that's a short list. So the classic example is one that we've actually talked about back in the metabolism chapter. Antibiotics that inhibit folic acid synthesis. Okay? Most bacteria can make their own folic acid. Humans can't. We have to eat it. Okay? Check the side of your cereal box, it will say folic acid right there. So, the two classic folic acid inhibitors are the sulfa drug, sulfonamide, and trimethoprim. These are very, very, very old school. Sulfa drugs predate penicillin. So, getting into the text that was on there that I'm skipping over. So this is the pathway for making folic acid, tetrahydrofolate. Okay? We as humans, we do not have this pathway. We don't have these enzymes. So organisms need folic acid to make a number of growth factors and nucleotides. Okay? It's a very important molecule for cells. So here we have two antibiotics used together showing synergism. Okay? This is your classic example of antibiotics boosting the effectiveness of each other. So we've talked about the sulfa drug before. Okay? When we talked about competitive inhibition of enzymes, we talked about sulfa drugs. Okay? Here's sulfanilamide. It goes by a number of names, like all pharmaceuticals. And this is the first precursor, PABA. They're so similar, the enzyme here doesn't know the difference. So enzyme 1 binds the sulfa drug, and it is shut down. But here's the problem. If you're using just the one antibiotic, there's still going to be some leaky expression. Not all the enzymes are necessarily shut down. There is tons and tons of copies of them. So it's making probably just enough folic acid to get by. So if you're taking sulfanilamide, you're also taking trimethoprim. Okay? It's prescribed as what's called Bactrim. Trimethoprim does the same thing, but to enzyme 3. So here they're working together, right? One stops the first enzyme, the other one's your backup to stop enzyme 3. It's just ensuring the pathway is fully shut down. Classic example of synergism. They're boosting the effectiveness of each other. All right, now the last class, there is the least amount of examples. Antibiotics that interfere with membrane integrity. Okay, so these are antibiotics that bind to the membranes of bacteria, interfering with their integrity, making the cells leak and burst. So here's the problem with these antibiotics. They don't get through peptidoglycan. Okay, so that would make them sound like terrible antibiotics. Well, yes and no. They don't do squat against gram-positives. Right? They cannot get through that thick peptidoglycan cell wall. But what about gram-negatives? Right? Very thin peptidoglycan surrounded by what? An outer membrane. Polymyxin B, topical cream, right? It's one of the 3 antibiotics in Neosporin. Polymyxin B is very effective against gram-negatives. Binds to the outer membrane, making them super leaky, causing them to burst. But the thing is that the membranes of bacteria are much like the membranes of humans. So this not only affects the membranes of bacteria, it also affects your epithelial cells as well. But fortunately, the top of your epidermis, most of the cells are dead. Okay, so polymyxin B you would not take orally. That would wreak havoc to your insides. So it's only used as a topical cream because it shows no preference to what membranes it binds to. So these are really good against gram-negatives, again, not particularly effective against gram-positives. Okay, so those are the 5 classes, right? Inhibit cell wall synthesis, inhibit ribosomes, inhibit nucleic acid synthesis, inhibit metabolic pathways, interfere with membrane integrity. So I left you folks thinking about one big thing. Is there any part of growth we haven't touched? Right? We got cell walls, transcription, translation, DNA replication, membranes, and metabolism. That's it, right? That's why, for the last 40 years, there has not been a new class of antibiotic discovered. We cannot figure out what else we can target. Every other year, though, for the last 6 years, I see some new promising antibiotic where they say, yes, this is a new novel class of antibiotic, and then something happens where it's not. Okay? There was one not too long ago. If it looks promising again, I'll post the article to Blackboard, but you have to remember to check. All right, so this last one is kind of a loose end. Sometimes they call it class 6. It's not really. Treating tuberculosis. Mycobacterium are so unique, right? We worked with them when we did acid-fast staining. They don't gram stain. They have that waxy layer around them, that mycolic acid. Tuberculosis is a very slow and chronic disease, very difficult to treat because of that waxy layer. Antibiotics have a very difficult time penetrating the cells. And 2, they grow so damn slowly that they can wait out the antibiotics. So what do you do to treat tuberculosis? Well, they first will put you on a cocktail of first-line drugs. 5 different antibiotics. And not for weeks, for months, if not longer. So in the cocktail, we've got streptomycin and rifamycin. Right? Streptomycin is an anti-ribosomal. Rifampin, rifamycin, whatever you want to call it, that's the one that causes red man syndrome, the anti-transcription antibiotic. Now, the other 3, though, are unique to tuberculosis. Isoniazid and ethambutol, those drugs are a select few that interfere with that waxy layer. Okay, so you can only take those for tuberculosis, maybe leprosy. And then the last one there, pyrazinamide, for the longest time, they didn't know what the hell it did. And I check these things like every year, and I was tickled that last year somebody finally sorted it out. Pyrazinamide causes the inside of the bacteria to become highly acidic, taking them out of their pH range. So this is what you would take for a long, long time if you had tuberculosis. This is ineffective. There is a second line, which is even worse. Okay, gotta make up some time here. So what we're going to look at next is bacterial resistance to antibiotics, because in nature, wherever there's an offense, there's always a defense. Right? If there's bacteria that can make antibiotics, there's bacteria that can combat them. So overall, we can look at resistance as either being passive or active. So passive or innate resistance is where something about the bacteria's structure just happens to make it resistant. So gram-negatives, for instance. Gram-negatives have that outer membrane. That is passive resistance. Okay? Many antibiotics have a hard time getting through that outer membrane. It's not there just for antibiotics. It's just kind of lucky. Right? It's passive resistance. It's a part of their cell structure that just happens to make them resistant to antibiotics. Now, the scary stuff, though, is active resistance or acquired resistance. Because in active resistance, this is where bacteria will have genes that allow them to react to and directly deal with antibiotics, circumventing them. And why it's often called acquired is because those genes can be passed around through transformation, transduction, conjugation, horizontal gene transfer. So let's see how they're active. Okay, how do they actively resist antibiotics? I'm skipping this one for now. We'll get to it actually in lab at some point. Okay. So the first example of active resistance enzymes that target and inactivate antibiotics. So these are exoenzymes. They are secreted out in response to an antibiotic. They target the antibiotic and they break it up. So the classic example, beta-lactamase. We're going to work with this in lab today. Okay, it's actually on the plasmid we're going to be working with, but beta-lactamase is an enzyme that does one simple thing to beta-lactam drugs. It snips that beta-lactam ring. Right? Because we went through all those generations of beta-lactams. They all had that square ring. That ring is important because if you disrupt it, it ruins the antibiotic. So, we've got penicillin, and we've got its inactive form, penicilloic acid. This will do nothing to a bacterial cell. Okay, so this is one example. There are other enzymes that can be secreted to disrupt antibiotics. This is kind of clever. Altering whatever molecule would have been targeted by the enzyme. So we can look at this in two different ways. You can change the shape of a protein that would have been targeted, or you can use a backup version. So in the case of ribosomes, right, class 2, where antibiotics had targeted ribosomes exclusively, some bacteria can react by altering the shape of the ribosomes so the antibiotic cannot bind. Right? That's pretty clever. Or, alternatively, and I'm going to have to make up time with this guy. Normally I talk a little bit longer about it. Penicillin binding protein. So MRSA is resistant to what? 4 generations of beta-lactams. Okay, so why is that? Well, the enzyme that makes the bridges between the layers of peptidoglycan here, we gave that poor enzyme the name the penicillin binding protein. We discovered penicillin before we determined the name or what this enzyme does. So, the penicillin binding protein is what gets inhibited when a cell is treated with penicillin. So what does MRSA do that's so different? It makes a different version of this penicillin binding protein. A version different enough that 4 generations of beta-lactams can't touch it. Only that 5th generation can. That's why it's so resistant. Just says, all right, I'm making a different version of this protein. Altering membrane permeability. This really only applies to gram-negatives. Okay, so gram-negatives can show resistance by effectively turning off their transport proteins. Okay, so they have porins, for instance. Porins let all sorts of molecules through that outer membrane. So a gram-negative can say, hey, there's an antibiotic around. I'm going to stop making porins, shutting out the antibiotic. So that's all they're doing. They're keeping the antibiotic out of the cell by walling it off. All right, now we get to the scariest form of resistance I know of, antibiotic expulsion. So this is actually something we talked about a while back. So in chapter 4, we talked about transport mechanisms, right? We had simple diffusion, osmosis, facilitated diffusion, active transport, group translocation. This is a form of active transport, efflux pumps. So this is a transport protein that acts as an antibiotic sump pump. Right? So at home, you get sump pumps in the basement that pump unwanted water out when it rains. So from an area of low concentration, your dry basement, to an area of high concentration, back out in the rain. Efflux pumps do the same thing, but to antibiotics. As they get into the cell, they get pumped right back out. Okay, multi-drug resistance here. So pump just about anything that gets in the cell back out. And then we got here, alteration of metabolic pathways. So if a metabolic pathway gets shut down, say that one for folic acid, a bacteria can react in a couple of ways. One, they can say, screw it, I'm just going to become an auxotroph and go find my own folic acid. That could happen. Or two, this is clever, some of them have detour pathways. So a hidden metabolic pathway that gets around the shutdown component. That occasionally happens as well. we got 5 mechanisms of inhibition and then 5 mechanisms of resistance, right? So we've got drug inactivating enzymes. We can change the shape of the targeted molecule. We can use a different version. We can have efflux pumps. We can alter membrane permeability. We can use detour pathways. Okay, so just clever ways of getting around what could be inhibited. All right, so second to last thing we're going to go through here, and like I said, I have to go a little long today. I want to briefly talk about how do we figure out which antibiotics are effective against which bacteria. And some of this we'll get to do in lab. So the test that you traditionally start with is the disc diffusion method, or what we're going to call in lab the Kirby-Bauer method. So somewhat familiar picture here. So we mentioned antibiograms in Chapter 10. This is an antibiogram. It's a Kirby-Bauer test. So you'll do this in lab. You're going to swab some plates with 6 different bacteria. And then you're going to place these little discs on the plates. Each disc contains a different antibiotic. Okay, so shotgun approach. You're throwing 10 different antibiotics at this organism. So you put these discs on, they go in the incubator, you come back the next day, 2 days later, and you look for the clearings around the discs. That's the important stuff. So these zones of inhibition. Okay, this big old clearing here. So on the first day, we're going to inoculate, we're going to put these discs on. The second day, we're going to bust out rulers, and we're going to measure those clearings. Because here's the correlation. The bigger the clearing, the more effective the antibiotic, right? You get this zone of inhibition, this no man's land, where the organism couldn't grow. But on the other hand, where you get something like this or this, that's probably not very effective, right? The organism grew right up to the disc. So that wouldn't be useful. I'll have a fun story when we do this in lab, but this is a clinical test that's still done very commonly to figure out what antibiotics still work against resistant organisms. Whoops. Yeah, we'll just skip to here. No, I won't. All right, so that's your first step. Okay, you figure out which antibiotics work, which brings you to your next test, broth dilution tests. So here we get a little bit more refined. Okay, we say we know this antibiotic works, but we want to know exactly what concentrations are effective. Fine-tune it. So you can calculate things like minimal inhibitory concentration or minimum bactericidal concentration. So was the least amount required to slow growth versus the least amount required to stop growth. So this is a modern take, and I've never done it this way. I may switch diagrams back at some point. But this is a more modern take on a broth dilution test. So this is how it works. We've got 5 different rows of antibiotics, right? Doxycycline, the sulfa drug, streptomycin, ethambutol, and kanamycin. So each row is a different antibiotic, but you set it up with decreasing concentrations. So you dilute the antibiotic from left to right. After you set up your dilutions, you inoculate the whole mess with some organism. If they're doing ethambutol, it's probably tuberculosis. So you incubate it, you come back, and what you want to know is where is their growth and where isn't their growth. So look at kanamycin and ethambutol. So you got growth in the first well. Okay, that concentration is not effective. You get growth in the second well. That concentration is also not effective. But look at that. In the third well, there's no growth. That tells you whatever the concentration is here, that's your minimum inhibitory concentration. So, I don't know, say 5 micrograms per mil. That is the least amount of antibiotic used to slow the organism down. So that would be useful in starting to determine size of dosage. You can see they're all different. So streptomycin here, no growth across the board. You don't know the MIC. You'd have to dilute it further to figure it out. Doxycycline at the top here is totally useless. It grew through all the concentrations. And then the sulfa drug, I don't know. I can't tell with these little spots here. Either the second to last one or the last one is the MIC. I don't know. I can't tell. The book doesn't say either. And then the last one is something I've never had a chance to do. I've always wanted to. Another kind of a modern twist on actually both of these tests. So E-tests combine both Kirby-Bauer with the broth dilution test, all in one. So instead of getting little discs with antibiotics in them, you get strips. So the strip here at the bottom, this has got vancomycin in it. Now, unlike the discs, which have a set concentration, you'll see how these numbers gradually decrease. That's the concentration of the antibiotic. Okay, from 256 probably micrograms per mil down to 0.016. So you inoculate your plate, you lay down your discs, you come back the next day. And you'll see that there's zones of inhibition there, right? You get clearings, but they're egg-shaped. Here's the neat thing. So this one ends at about 2. Wherever the clearing ends, that number is supposedly the minimal inhibitory concentration. So here, vancomycin's effective at 2 micrograms per mil. Right? And you can see in the top diagram, all the egg shapes are a little different. Mostly. So that's pretty neat. Combines both tests in one. Alright, my last few loose ends here, and then I'll be quiet. I'm sorry. So up to this point, we've tackled nothing but antibacterial antibiotics. Right? We spent all that time talking about how to inhibit different parts of microbial growth. And that's most of our antibiotics. So what about dealing with, say, viruses? That's a whole different ballgame, folks. Antiviral drugs are totally different from antibacterial antibiotics. So, for instance, most antiviral drugs are man-made. And they are all very different from each other. Okay, they are designed to target various viruses. Or specific viruses, sorry. But all antivirals basically do similar things. They inhibit different steps of the viral replication cycle. Right? You have some that inhibit attachment, penetration, biosynthesis, maturation, release. So if you can screw with one of those steps, you can slow down the virus. So I got a whole list of them here, but let's just look at the diagram. So treating HIV. So these days, individuals with HIV are given a cocktail of antibiotics in a single pill. Antibiotics, antivirals. And here we're illustrating which steps in HIV's replication cycle does each antiviral inhibit. And I can never say these names right, so I'm sorry. Let's see here. So, enfuvirtidine, it's like a Latin one. This one prevents attachment. Let's see, we get ritonavir. This one inhibits the proteases it would make to start breaking down your cell's proteins. Let's see what else. Raltegravir. That one sounds Scandinavian. This one prevents the virus from becoming a prophage, an integrase inhibitor. Let's see what else. AZT. AZT is super infamous. So, I don't know if anybody's ever seen Dallas Buyers Club. Fabulous movie. That whole movie's about AZT. Okay. They didn't know we should be giving people cocktails of these drugs and at what dosages. So, AZT at high concentrations is a little dangerous, but it is a highly effective reverse transcriptase inhibitor. So it prevents the virus from turning from RNA to DNA. And I think that's all of them. But just showing you that each antiviral very specifically targets different steps of just HIV's replication cycle. Okay, you want to deal with, say, the flu, totally different antiviral drugs would be used. Antifungals, I'll be super brief here. When dealing with fungal pathogens, you got a problem. They're eukaryotes. So are we. So the differences in what you would target become more muddled, more gray. So you have to find something unique to fungus to target. So for instance, ergosterol. All membranes have some form of cholesterol in them. Okay? Fungi just happen to have a unique form of cholesterol called ergosterol. So drugs like polyenes and azoles, they interfere with ergosterol formation, causing the fungi to become weak and explode. So you can see that yeast cells bursting. Oh, and most of these are given topically. If you have to take systemic antifungals, you're in for a bad time. All right, and then the last one here, real quickly. Antiprotozoans and antihelminthics. So dealing with protozoan parasites like Giardia or trichinosis, or, I don't know if you folks know what a helminth is, dealing with worms. So there's all sorts of examples. I just have two here. So if you say you have Giardia, you went camping, you drank, unfortunately, a still pool of water, and now you have explosive diarrhea, they will probably give you metronidazole, Flagyl. Flagyl is actually very effective at killing intestinal parasites. It disrupts their DNA, so they cannot easily copy their chromosomes. Fun fact, it's actually useful against obligately anaerobic gram-positive bacteria, if I remember correctly. But it does have some unfortunate side effects. It is mildly carcinogenic, it can and will cause birth defects in pregnant women, and it disrupts your blood cells. So you can see the individual's got black hairy tongue. It's just from the broken down blood vessels. Dealing with worms, from what I have read, the most popular choice is mebendazole. Mebendazole interferes with the cytoskeleton within their cells. So, for instance, if you had a tapeworm, you'd take mebendazole. And mebendazole would slow or lower the tapeworm's ability to absorb nutrients from you. Okay, so after a few days of mebendazole, you'll find a very long and unfortunate tapeworm in your feces because it starved to death. Okay, I'll end on that fun note. So, folks, I posted the quiz for this yesterday.